Hello, today we're going to be discussing the plasma membrane structure and its many functions. The goals today are going to be to be able to understand the fluid mosaic model of cell membranes, describe all the different functions of phospholipids, proteins, and the carbohydrates in the membrane itself, discuss membrane fluidity, and explain passive, tonicity, and osmosis and diffusion. We're also going to be able to understand at the end of this lecture what active transport is. Okay. The last lecture we talked about the interior of cells and their organelles and their functions. Before we start getting into the plasma membrane, we have to talk about the exterior of cells and what is going on the outside of the cell. Okay. Number one, in animal cells, there is no cell wall. They do, however, have an extracellular matrix. This is pretty much a meshwork of different fibrous proteins and intermediate filaments and polysaccharides. It does contain also elastin and collagen, which allows for it to be flexible, give it some sort of movement, but the matrix does vary according to what cell type it is. So if we need it to be a little bit more hardened in bone cells, then that's what the matrix is going to be. In animal cells, there are 3 different types of cell connections. Okay, this is the way that cells actually communicate amongst each other. The 3 types of cell communications are tight junction, adherent junctions, and gap junctions. So tight junctions are when 2 cell plasma membranes are actually fused together very tightly. So it's pretty easy to memorize. Tight junctions, plasma membranes fused very tightly together. Reason being, they actually do not want to allow any ions or molecules to go through them or between them, because they do not need those types of particular ions or molecules to be transported into the cell. So they'd rather go ahead and just make this huge tight junction and only allow very small or specific molecules through, such as water. Gap junctions, however, are literally, just imagine, kind of like channel proteins. Okay, they're going to allow the cells to communicate amongst each other, and you can see right here in this channel protein right here, this gap junction is there's a gap in between the cell plasma membranes, but there's these channels that actually allow them to communicate and transport molecules, ions, or other substances through as needed. Adhesion junctions, okay, this is when cells are fastened to one another and to the basement membrane of one another, and this allows for sturdy but flexible sheet to be actually obtained between the cells. And as you can see, molecules and ions can still pass through in between these 2 plasma membranes of these 2 different cells. So it does want to allow a lot more molecules through so that potentially they can fuse across the plasma membranes of each of these cells, but it's not going to be as tightly wound up or tightly adhered to each other as the tight junctions. Okay. Now, plasma membrane, what is its structure and its function? Okay, so sorry about that. It is selectively permeable, which means it does only allow certain molecules of ions through it. Because remember, your DNA is inside of the cell, so your plasma membrane is they're kind of like bodyguards. They're not going to let everything through, because potentially some things can damage your DNA inside of the cell, and if that damaged DNA gets replicated, it could potentially cause things such as cancer. So it is only going to allow a couple of items through that should be there. So what makes up the phospholipid bilayer of your plasma membrane? Well, we do have a phosphate group and a glycerol attached to a glycerol. These both make up your hydrophilic head. Hydrophilic means it's water loving. But then you also have these fatty acid tails, which are actually sitting here and attached to your hydrophilic head. These guys, though, are hydrophobic. They don't like water. So this is how they arrange themselves in the phospholipid bilayer. Okay, we have the hydrophilic heads right here. Okay, these are polar. They like water. So that's why they're facing the outside of the cell and inside of the cell where there is water. These hydrophobic tails do not like water. They're nonpolar. That's why they face one another, so that they don't ever have to be exposed to the watery material outside or inside of the cell at all. Okay, the phospholipid bilayer is embedded with different kinds of proteins throughout. Right here, all these little blue turquoise looking things, these are proteins. Okay, and the plasma membrane is a fluid mosaic model. They say it's fluid mosaic because it does have quite a bit of movement going on, which you will see in the next slide. Go ahead and watch this video. It's awesome because it actually shows you how fluid the plasma membrane can actually potentially be.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions.
So first, let's go ahead and talk about the structure of the plasma membrane. We just saw some proteins that are embedded throughout the plasma membrane right here. And this is also the hydrophobic membrane region. All this right here, remember, because this is the fatty acid tails. They do not like water, okay? The fluidity of the membrane, remember, it's called the fluid mosaic model. It's actually showing you, just like in the video, that these phospholipids actually are never ever just stable and they're not stuck in place, okay? They're going to have some lateral movement, which is, you can see right here where these guys actually flip, okay? They can actually flip, okay, at certain times, and also they can actually do a flip-flop, which means this one that's facing the exterior of the cell can flip with this one and actually now face the interior of the cell. This happens about once a month, okay? But they are fluid and they are moving, okay? What does this affect, okay? These, the fluid mosaic model is actually affected by these actual tails, these fatty acid tails that are on each of these phospholipids, okay? Right here, this is an unsaturated fatty acid tail because it has that kink, and this is a saturated fatty acid tail because it does not have a kink or that double bond creating that kink, okay? This actually allows for fluidity of the actual plasma membrane, okay? Because it's actually going to allow it to be very fluid and allow things to come through and so forth that need to come through. And also it allows for, it actually accounts for the viscosity of the plasma membrane as well, okay? That's just the amount of structure and the composure of the actual plasma membrane and allowing things to come through or not through, because obviously with the saturated hydrocarbon tails right here, a lot of things are not going to be able to come through, such as over here with the hydrocarbon tails that are unsaturated and have that kink, okay? What is the effect of the cholesterol that is embedded inside of the plasma membrane as well? Okay, the cholesterol is actually allowing for structure and a little bit more of a stronger structure to be maintained with the plasma membrane itself, okay? Without the cholesterol embedded in the plasma membrane, it wouldn't be as strong as it is with it. And we talked about, or we actually saw, those different kinds of proteins sticking out of the plasma membrane at the very beginning. But there's different kinds of proteins. If it's considered an integral protein or integral, okay, then that means that it's going to be going from one side of the plasma membrane all the way to the other, okay? So for example, right here, it's going to go from here to here, from the exterior part of the cell membrane all the way to the interior. It comes completely across through the actual plasma membrane from one side to the other. These are integral proteins, okay? And we'll go ahead and see a picture in the next slide right here of an integral protein. As you can see, this is actually the protein itself. where it's coming all the way through and across the entire plasma membrane. Peripheral proteins, on the other hand, do not actually reach completely across the plasma membrane, okay? So remember, this is an integral protein, but these peripheral ones, like this one right here, is only found either on the inside or the outside of the plasma membrane, but not throughout the plasma membrane. It does not go from one side to the other. So another peripheral, let's say we draw one right here, that would be a peripheral membrane on the inside of the cell. If we had a peripheral membrane, let's say right here, let's say it was over here on the top side, facing the exterior part of the cell, but it did not embed itself. It is not throughout the entire plasma membrane. Okay, it does not reach across fully. That is a peripheral protein. And then we have the different functions of these proteins that we're talking about. Okay, some proteins allow for transport to be done. Some allow for enzymatic activity to occur. Some proteins are going to actually allow for a signal to be transmitted across from the outside of the cell into the inside of the cell. Okay, some of these proteins are helping with cell-to-cell recognition. Some are also helping with intercellular joining. Okay, these are functions of different proteins. Okay? Attachment to a cytoskeleton and to the extracellular matrix is also a function of proteins that can help with the structure and the formation to be maintained. Okay, so let's go ahead and take a look at these guys individually. So the first one we're going to be looking at is the cell-to-cell recognition proteins. Okay, as you can imagine, it's literally allowing one cell to identify another as either foreign or a part of itself. Okay, this allows for our body to distinguish what should be killed and what shouldn't be killed. So if there's an foreign invader like various viruses or bacteria, that's your cue, or that's actually your key for your body to go after it and kill it and get rid of it before it can do any damage to your actual DNA. But cell-to-cell recognition proteins actually allow your cell to identify one cell and being okay and being like, hey, that's just another cell. Look at the flag I'm carrying. I'm a part of the body. I'm a part of this part of the of your body. Don't kill me. That's what it's doing. It's potentially just letting other cells recognize it as not a foreign invader. Okay. Receptor proteins. Okay. These allow signal molecules to bind and then causes a cellular response on the other side of the cell or maybe somewhere down the line. So in other words, right here, this substrate can potentially bind right here to this receptor protein, and then it's going to either release a signal down through here or maybe down the row over here at another part of the plasma membrane. But that's the whole point of the receptor protein, is to recept and then allow some sort of transmission of information to the inside of the cell. There's also enzymatic proteins. Enzymes, as you all know, actually help chemical reactions occur quicker because they reduce the amount of activation energy required for that chemical reaction to occur. Okay, right here is an enzymatic protein, meaning that a substrate can sit here and bind to it and then potentially release a different kind of product that your cell was needing at that time. There's also junction proteins. We kind of just looked at a couple of the adhering, the gap junction channel proteins a little while ago. Okay, so junction proteins just form between cells and cells and allow for cell-to-cell adhesion and communication. So good example was the channel proteins that we looked at a little while ago, and the gap junctions that were formed because of them. So now we're going to talk about transport proteins. Okay, so transport proteins, what are its purposes? What does it do? As you can imagine, transport, okay, it's actually just going to be responsible for allowing the transport of molecules or ions into or out of the cell itself. Okay, examples of transport proteins would be channel proteins or carrier proteins. Right here, you can actually see this channel protein up top. It's literally just kind of like a tunnel that's formed that allows some ions or molecules through to the inside of the cell. Carrier proteins. This is a carrier protein right here, the purple one. It's going to allow molecules or ions to attach to it and then ultimately be released on the other side. You all have heard of active transport and passive transport. Okay, we're going to start first by talking about passive transport, because these are three Great examples of passive transport that you should be able to recognize or know about: diffusion, osmosis, and facilitated diffusion. Okay. Passive transport is passive because it does not require any kind of energy input at all. Okay. On the other hand, we have active transport. Energy is going to be required to move these substances, ions, or molecules across the plasma membrane. So that's a huge difference. Active, you need energy. Passive transport, no energy is required. So we're going to start first by talking about diffusion. Okay, this is where molecules of any substance are able to spread out evenly into some sort of available space. An example of this is, let's say you put cologne or perfume on before you enter a room. When you enter the room, the people closest to you will slowly start to smell that cologne or the perfume, because the molecules are kind of basically trying randomly to spread out throughout the entire room until it's equaled out and spread out evenly throughout the entire room itself. So right when you walk in a room, those, like I said, the cologne or perfume is starting to spread, so the people closest to you will smell it first, and then the people that are farthest away from you will eventually smell it once these molecules have spread out throughout the entire room. So diffusion, we're talking about the actual molecules or the substrate or the solutes themselves dispersing across, okay, a permeable membrane. So it's a membrane where larger molecules and ions can diffuse across. So right here, it's going to go from a high area of concentration, because there's a lot of orange molecules on this side, and it's going to diffuse across as time goes on, because these guys are moving randomly. They're just kind of jumping around, and eventually they're going to make it across this membrane and completely reach equilibrium, where they're equal out on both sides. When they reach the state of equilibrium, that does not mean they stop moving. They continue to move around, but they will continue to go back and forward, because it has already reached equaled out on both sides. So it has reached the state of equilibrium at this point. Okay. Right here, we're going to be pointing out substances, and this is diffusion still. Substances are diffusing down their concentration gradient. So they're going from a high to a low concentration. Okay. So right here we have a high orange molecule concentration on the left, and then a very low orange molecule concentration on the right, because there's actually none over here. So what's going to happen, like I said, these guys are randomly moving, and the purple ones are randomly moving. So the orange ones are going to continue to randomly disperse until it reaches a state of equilibrium, and at the same time the purple ones are going to go from a high to a low concentration on the other side of the plasma membrane. So what's happening, they're going to even out eventually till they both each reach their state of equilibrium. So diffusion is the movement of the actual solutes or from their high to their low areas of concentration. When we talk about osmosis, we're actually going to be talking about the movement of water across a semi-permeable membrane. Okay. In diffusion, remember, it was a permeable membrane because larger molecules and ions can transfer across that membrane. In osmosis, it's actually semi-permeable because it only allows a few things across, and in this case, we're going to be looking just at water. Okay, it does depend on the solute concentration, though, however, and it actually maintains osmoregulation, which is the control of the water balance from the inside of the cell and the outside of the cell. So that's what's actually allowing for water balance to be maintained and controlled. Okay. To understand what's going on with osmosis, we can start talking about tonicity, which is the ability of a solution to cause a cell to gain or lose water. Remember, gaining or losing water is the movement of water molecules, which is osmosis. Okay. Tonicity is going to have a greater impact on the cells without cell walls. So that means it's going to have a greater impact on mononuclear cells and animal cells, but not so much an impact on plant cells because they have a cell wall. So tonicity, right? This is what's happening when we put these cells, these red blood cells, into a hypertonic environment. Hypertonic means that the solute concentration outside of the cell is greater than inside of the cell. So for this example, let's say this is a beaker and this is my red blood cell right here. We're just going to look at one of these. So this is my red blood cell, let's say for example right here. So if it's in a hypertonic solution, that means that, let's say my solution inside that beaker is 50% sodium chloride, okay? And then my concentration inside one of these cells is, let's say, 1% sodium chloride, okay? That's what it means by you're putting that cell in a hypertonic solution. The solution of the solute concentration is higher outside of the cell than inside the cell, okay? What happens when you do this? Well, as you can see in the picture, the water is actually moving from inside the cell to outside the cell because it's trying to even this part out. That's the only way it can equal these guys out, because the solute cannot move, so only the water can move across. So if you have 1% of sodium chloride inside the cell, that means you're going to have the remainder being water, so 99% of water is inside the cell. So water is going to go from inside the cell to outside the cell to try to equal them out. If you put a cell into an isotonic solution, that means the solute concentration is equal both inside and outside the cell. So let's say the red blood cell right here has 50% sodium chloride, and then outside in this beaker, let's say for example, there's 50% sodium chloride. So the molecules of water are still going to transfer back and forth, but it has already reached a state of equilibrium, okay? And last but not least, we have a hypotonic solution. Here's my beaker, here's my red blood cell. Hypo meaning less than. So let's say inside the cell we have 10% sodium chloride, okay? And then that would mean outside the cell, let's say there's only 1% sodium chloride solution, okay? So what's going to happen is right here the water, which is 99% outside the cell, is going to be moving inside the cell to try to go ahead and equal these guys out. It's going to add more and more water till this eventually becomes 1%, just like the outside. It wants to reach that state of equilibrium, okay? But the problem with this one is when you put a cell into a hypotonic solution, it's going to fill and fill and fill inside of the red blood cell until it finally bursts, because there's nothing protecting it from getting too swollen here. So it will eventually burst in the animal cell. But plant cells and fungi have cell walls. These actually help to maintain this water balance, okay? In animal cells, if you put a plant cell inside of a hypotonic environment, okay? Hypotonic, it's meaning less, there's less solute outside than inside the cell, okay? What's going to happen is water is going to fill inside of the cell because it's trying to, remember, reach equilibrium. But the only difference is in the cells, with plant cells, it's never going to burst because you have that cell wall protecting it from bursting. Isotonic solution, water is moving in and out of the cell itself because it's equal outside and equal inside according to the solute concentration. And then if you put a plant cell into a hypertonic solution, what's going to happen is the solute concentration outside the cell is greater than inside of the cell, so water is going to move outside to try to equal it out, okay? This is what's called plasmolysis. When a plant becomes very shriveled and kind of weak, if you leave a celery stalk out for a day or two, it becomes plasmolized, which means plasmolysis has occurred. In other words, water has left inside the cells to outside of the cell, okay? This is the comparison, so you can look at animal cells and plant cells at the same time when trying to understand tonicity, okay? So right here is in if you put the red blood cell in a hypotonic solution, it will eventually burst because water is going inside. Same thing right here with the plant cell. If you put it in a hypotonic solution, water is going inside, but it never bursts because you have the cell wall protecting it from bursting. In an isotonic solution, water is going to move inside and outside of the cell in a red blood cell. And right here as well in a plant cell, it's just moving inside and outside because it's already reached equilibrium state. If you put either of these guys in a hypertonic solution, water is actually going to go from inside the cell to outside the cell. It's going to go from a low solute concentration to a high solute concentration because it's trying to even it out, remember? So right here, the animal cell becomes shriveled up And as well, same thing happens in a plant cell. The plant cell will become shriveled up, and this is called plasmolysis in plants. So now that we talked about diffusion and osmosis, we're going to go ahead and talk about facilitated diffusion. Okay, facilitated diffusion is still passive transport, and no energy is required here. And what it does is it actually contains transport proteins that speed the movement of molecules across the plasma membrane down their concentration gradient. Okay, these are considered transport proteins, and we talked about those a little while ago, which are the channel and the carrier proteins. So let's go ahead and look at a picture of all of these. Okay, so remember we started first by talking about the diffusion of molecules. They go from a solute high concentration to a solute low concentration. Okay, and we also talked about osmosis, which is the movement of water from a low solute concentration to a high solute concentration. And then we have now the facilitated diffusion, okay, which are carrier proteins and channel proteins. This one on the left right here would be an example of a channel protein because it just kind of makes like a tunnel. It allows that molecular ion to pass through. No energy required at any of these. And then here would be my carrier protein, where the ion or molecule attaches inside of the actual protein and then gets dispersed onto the other side of the plasma membrane. On all of these, including osmosis, there is no energy required. Now we're going to go ahead and start talking about active transport, which, as you can see, does require ATP, which is energy. So active transport does require energy. Okay, it's going to move solutes against a concentration gradient, and a good example to understand and know is going to be the example of the sodium-potassium pump. So first and foremost, the exterior part of the cell does have this positive net charge, and it is across the membrane potential right here that it actually has this chemical negative charge inside of the cell. So as you all know, positives are attracted to negatives and negatives are attracted to positives. So what does these sodium ions actually want to do? These sodium ions want to be pumped into the cell where there's a negative charge. Remember, positive is attracted to this negative. Okay, that is what wants to happen. And at the same time, these potassium ions, they're happy. They should be happy right here with this negative charge because positives attract this negative. Well, what's actually happening? Okay, let's talk about what's actually occurring. Okay, so 3 sodium are pumped outside of the cell. Okay, so we're going to have 3 of these guys going. Remember, they're positively charged. They're going from this negative charge and they're going to be pumped out, okay, into this positive environment. So we're actually moving a positive towards another positive environment. And for every 3 of these sodium that are pumped outside of the cell against the membrane potential gradient, we also have 2 potassium pumped into the cell. Okay, potassium, they are positively charged and they actually want to go towards the negative charge right here. They're going to be pumped into the cell. Okay, this requires energy though, because if you recall from the last part where we were talking about diffusion and osmosis, ions and molecules normally want to go from an area of high concentration to an area of low concentration. So these potassiums should want to go from this side inside the cell to outside the cell, but that's not what's happening. They're actually going against their concentration gradient. Okay, they're going against it. They're actually going from this, let's say 1% of concentration outside the cell to the inside where there's already 99% concentration. So they're doing the opposite of what we've been talking about. They're going from an area of low to an area of high solute concentration. Same thing with the with the sodiums down here. Those are what the potassium are doing. Now the sodium, same thing. It should potentially want to go from high area of concentration to an area of low, but it's doing the opposite again. This is actually going from a low solute concentration to an area of high solute concentration. Okay, and it's also going against its membrane potential because it's going from it's positive. Remember, sodium is positive and it's on this negative charge over here, but it's actually going from a positive to a positive charge. So this is against the electrochemical gradient of the plasma membrane, which is why this is what's requiring energy, unlike the passive transport that does not require energy. So, what is the membrane potential? It is an electrical potential energy due to the separation of opposite charges. Remember, exteriorly, we actually had a positive charge, and interiorly of the cell, we actually had a negative charge. Okay, that's what is creating this membrane potential. There's a chemical difference, and this is just describing the two charges I just explained. Okay, electrical chemical gradient, which is what we discussed right now with the sodium potassium pump, is caused by the concentration electrical gradient of the ions across the membrane. So this is potentially causing both of those to have what's called an electrical chemical gradient. All these positives, all these negatives, okay, that's what it's causing. Right here, you can actually see a proton pump, but we actually have energies required once again, because you can see this electrical chemical gradient occurring because here is hydrogen ion, which is positively charged, and it's on the negative side of the cell. It's on the inside. But instead of saying over here, it's going to be going towards another positive charge. Okay, so positive is going towards positive. It requires energy, okay, because it's not normal. Usually it should want to stay on this side where the negative is. Okay, so an electrogenic pump is a transport protein that generates voltage gradients across the membrane. And like we discussed, an example of that would be the sodium potassium pump in animals. There's also the proton pump, which occurs in plants, bacteria, and fungi as well. Okay, this is what's responsible for actively transporting hydrogen ions out of the cell. And hydrogen ions are important to keep in mind, because remember, that's potentially what pH is. pH is measuring the amount of free hydrogen ions. And the more free hydrogen ions you have, the more acidic your actual cell or your substance will be. Co-transport proteins, what they do is, let's say we have the proton pump we were just talking about right here in a plant cell, where the hydrogen ions are getting transported across. Well, and it's releasing these hydrogen ions that are free on the exterior part of the cell, and that is going to actually drive the passive transport of another solute. So here's active transport. Active, it's active because it's requiring energy input. And then that is actually going to be pushing and helping this passive transport occur as well. So what's happening is this is actively pushing those protons out and help these hydrogen ions to allow them to be free to help with co-transporting or co-assisting the transport of this sucrose back into the cell. So there's a reason for them to be pumped out. And bulk transport are when you're talking about moving large molecules, okay? There's different kinds of bulk transport. There's exocytosis. This is the transport of vesicles from the Golgi apparatus, okay? It releases their contents out of the cell. Endocytosis would be basically the uptake or the intake of macromolecules into the cell. So exocytosis, exit, it's basically a bulk transport of items exiting the cell. And then endo, you're going to actually be allowing things and taking things into the cell. So there's different types of endocytosis. Remember, endo, taking it into the cell. Okay. The phagocytosis is the engulfment or a large chunk of the actual macromolecules to be engulfed. So it's like a huge big bite of whatever it's trying to actually allow into the cell. That's phagocytosis. Pinocytosis is a tiny little pinch, so basically a tiny pinch of or amount of the actual macromolecule coming into the cell. So phago is a large engulfment, pino is a tiny little engulfment of items coming into the cell, and there's also a receptor-mediated endocytosis. That just means that you have to have a receptor that recognizes the contents that are coming into the cell, and that receptor flags it down, says, hey, come over here, this is where you need to enter, and then it flags it down, and then those contents are transported into the cell due only to that receptor that mediated the process though. Okay, and here are some really good examples that you should practice. An artificial cell consisting of an aqueous solution enclosed in a selectively permeable membrane has just been immersed in a beaker containing a different solution. The membrane is permeable to water and to simple sugars such as glucose, fructose, but completely impermeable, which means it's not going to allow any of the disaccharide sucrose across. Okay, so what is that telling us automatically? Water can go across, as well as glucose and fructose. So automatically, I'm going to automatically unline my glucose and my fructose because I know these guys are the ones that are going to be allowed into or out of the cell according to its tonicity. Inside my cell, I have 0.03 molar of sucrose, a 0.02 molar concentration of glucose. That's inside my cell. And then outside of my cell in this beaker, I actually have the 0.01 molar concentration of sucrose, and so forth and so forth. That's the setup. So now think about what's going to be happening with each of these different simple sugars, as well as with water. What can we expect to happen? Question is, which solute will exhibit a net diffusion into the cell? So which one will actually allow one of these solutes, which of these solutes will actually go into the cell? Okay, diffusion. So here, remember, it allowed glucose and it's allowing fructose across the membrane, but sucrose cannot go across the membrane. So think about what happens with each of these. Diffusion is the movement of a solute from a high solute concentration to a low solute concentration. Okay, so let's look at glucose first. Glucose, is it higher in the cell or higher outside of the cell? Okay, it's higher inside the cell. Okay, so that glucose is going to try to even itself out by going outside of the cell. Okay, I'll just put glucose from an area of high glucose concentration to an area of low glucose concentration. Now let's look at fructose. Fructose, there's none inside the cell, so it's 0% molarity inside the cell, but it's 0.01 outside the cell. So the diffusion of fructose, where is it going to go? Remember, diffusion goes from an area of high solute concentration to an area of low. So that fructose is going to go from this high outside the cell to inside the cell to try to even them out. Remember, sucrose cannot go anywhere at all because it's not permeable according to this membrane, because that's what this question told us. So now we just discovered which solutes will exhibit a net diffusion into the cell. Well, our answer there would be fructose.